Alright, we're going to talk about Chapter 41, which is the nervous system. When we talk about the nervous system, it has two regions. It is the central nervous system, which is your brain and spinal cord. And it is your peripheral nervous system, which consists of your cranial nerves, spinal nerves, and the autonomic nervous system. Then when we talk about the actual central nervous system and peripheral nervous system, there are two types of cells. There are the neurons, which receive impulses and send them on to other cells, and neuralgia, which protects and nourishes the neurons. The other thing that we want to talk about is action potentials. The chief regulators of the membrane potential are sodium and potassium. Take us back to fluid and electrolytes. Sodium is the major positive ion in the extracellular fluid, and potassium is the major positive ion in the intracellular fluid. So remember potassium, intracellular fluid, sodium, extracellular fluid. So in response to the electrical stimulus, the cell membrane becomes permeable to sodium, moves into the cell, then its polarity of that cell membrane and the neuron is what they call depolarized. What happens then, that stimulates the action potential, which is the nerve impulse. The other thing that we need to talk about is the norepinephrine. This is where you have excitatory or inhibitory, and it is a major neurotransmitter. When we talk about norepinephrine, we can also talk about antinergic receptors, which are divided into alpha and beta. That took us back to cardiac, also takes you back to form. So remember that beta-1 receptors are found in the heart. You have one heart, and they are your receptor cells that regulate the rate and the force of the contraction. Beta-2, because you have two lungs, are the receptor cells of the lungs, the arteries, liver, and uterus. They also regulate bronchial diameter, arterial diameter, and glycogenesis. So that was just kind of a little bit of a recap for that part. This is a box from page 1409 in your textbook. It's the general functions of the four regions of the brain. This is a very important box to know that the cerebellum, for instance, function is to control skeletal muscle activity. You need to be able to differentiate what region controls what function. That's a very important part when you're assessing the nervous system. So please look over and review and study this box. I do love this picture from your textbook. I'm a more visual, hands-on kind of person, so I like one that it is color coordinated in different colors and that it is labeled. So this is just another way of learning where the lobes are and then some of the things that are affected based on the lobe. This plays an important part when you are assessing the nervous system to know, or if a patient goes for a scan of their brain and they have a tumor or something in the frontal lobe, that could impair their speech, versus where if it's in the occipital, they could have more visual type disturbances. The other thing I want to add on page 1410, there's a box, table 41-2. It's functions of the lobes of the cerebrum, and areas of the cerebral cortex. This is also where it's telling you the different areas and the functions, so it correlates with this picture. Then the other thing I want you to know is off of that page, it also has another table regarding cerebral spinal fluid. This is a procedure where they can actually go in and draw out spinal fluid and send it off and have it tested. You need to know that normal spinal fluid is clear and colorless. There's no distinct color to it at all, and it should not be cloudy or anything to it. So that's just another thing I would like for you to look over, is those two boxes. So now we're talking about the blood-brain barrier. The thing that you need to know about the blood-brain barrier is that lipids, glucose, amino acids, water, CO2, and oxygen can pass. It is a controlled environment. It is there to protect your brain, so not everything can pass through the blood-brain barrier. This is a defense mechanism for our body. We don't want just anything being able to cross that barrier into our brain, because then we have far more damage. So as you guys probably realized from pharm, there are a lot of different substances, such as antibiotics, that can't pass the barrier. Chemotherapy does not pass the barrier, so if you have patients who have brain tumors, chemotherapy usually does not work for them to get rid of the actual tumor. They usually have to have radiation because of that blood-brain barrier. The other thing that cannot pass through there is urea, creatine, proteins, and some toxins. Now, on the other side, if you have an injury or an infection of the brain, you can increase the permeability of the brain barrier, and it alters the concentration of proteins, waters, and the electrolytes which are there. This is a picture of dermatomes. Dermatomes are used as an assessment tool. For example, a patient goes to OR and they're given general anesthesia. When they come back, essentially their body has been kind of put to sleep and relaxed. So you need to assess the sensation and function of these dermatomes. So you will see these graphs, and they're used as documentation methods for, the patient came back and I assessed S1, they had sensation up to from S1 to L1, or they just came back and they only have sensation at S1. So it's used as a documentation tool, plus you can see this is also color coordinated, so it goes over your cervical, thoracic, lumbar, and sacral nerves. So this slide is your deep tendon reflexes. We've talked about them in the endocrine chapter. We also, I think they popped up in fluid and electrolytes. So just a few things to add on, or just kind of a refresher, is that the deep tendon reflexes are in response to muscle contraction, and they cause muscle relaxation. They also depend on an intact sensory and motor nerve roots, functional synapses in the spinal cord. They are also looking or depend on neuromuscular conjunction and a competent muscle. So everything has to be working. There should be no discrepancy, no deficiency in that muscle contraction relaxation system. If there is an abnormal DTR, it could indicate health problems or a lesion on the spinal cord. So with the next couple of slides, it's the sympathetic nervous system, the autonomic nervous system. This is also in your textbook in bullets on page 1414. You guys can review over some of the things that you would see if you have stimulation of the sympathetic nervous system or autonomic nervous system, so you guys can review over those on your own. This box is a very important box. I can't stress that enough. It is your cranial nerves. They are testing 1 through 12. There are 12 cranial nerves. They each have a function and a name and a number. You are responsible for knowing the number, the name, and the function. And when we get to the assessment techniques, you're also responsible for knowing how you would assess each cranial nerve. This is a big part of the neuro chapter, and you guys will practice assessing the cranial nerves in lab, but please look over that beforehand so that you are familiar with it. So this box is on page 415. The next couple of slides is the same box, it's just spread out.